Darren Link, co-founder of RainDance Technologies and Vice President of Research and Development. RainDance Technologies, what we're doing is we're putting reagents inside of droplets. We encapsulate the biology in droplets, one droplet at a time. And what's really important is to be able to do this in a way where we can put single cells, single proteins, single molecules of nucleic acids, individual reactions, one at a time, thousands per second, even up to 10,000 per second and beyond. And in doing that, we can look at single molecules, single protein reactions, single cells, and really get at the core and at the heart of the biology. The way the droplets work within the system is that we start out with a sample. Samples 10 microliters, 25 microliters, 50 microliters. We divide that sample up into partitions. Those partitions, it might be 1 million partitions, 2 million, 10 million partitions. And then it's individual reactions inside of those individual partitions, just like samples in a test tube. So we can add to those reactions, we can initiate reactions, we can even sort through reactions and select individual reactions from a collection. This is the processing. When you're done, you've analyzed that entire sample in incredible detail by looking at it one small partition at a time. Droplets are being used to solve biological problems in life science research in a number of ways. We have solutions for researchers in targeted sequencing and in digital PCR. In the targeted sequencing side, we have two instruments. One for low to medium throughput called the RDT1000, and we have a high throughput instrument called Thunderstorm instrument. The way these systems work is the researcher, they tell us, these are the regions of the genome that we're interested in. Maybe it's 1,000 regions, maybe it's 10,000 regions, maybe it's 20,000 regions. It's exactly the regions that they want, and they want to be able to do the sequencing on. We design PCR primers against those regions and set up the reactions, inside of the droplets. Part of what makes the technology special is the automation, the simplicity, the way the researcher can walk up to the instrument, load their samples, and confidently come back to have them completely processed. That's part of it. And of course that leads to lower costs, ease of use, the standard things that you'd be looking for in an instrument. But what makes it special to me is the performance. It's the performance of being able to have incredibly precise droplets, uniform in volume, and also incredibly precise in terms of the way it's processing the samples, the numbers of droplets, and getting into the millions of reactions. This allows you, when you're looking at for sequence enrichment, when you're looking at doing that, you enrich uniformly. Things that are of low prevalence in the genome show up at low prevalence. Things that are at high prevalence show up at high prevalence. You're able to see, minor mutant DNA in the background of wild-type DNA when you're doing cancer analysis. This is also incredibly important and true with respect to the digital PCR case. So between the automation and the ability to get very precise answers, this makes these systems, just very unique over what else is available and possible, and it's really enabling our customers to do things that just couldn't be done before.